This is Audiomunity, a podcast about our body's never-ending fight with the outside world. Hello, this is Audiomunity, episode, what are we on, 36? 35?
I think 36.
36. I'm Kevin Bonham. We're recording on March 11th, and I'm joined as ever by Matt Woodruff.
Hi, everybody. Good to be here.
And Kate is once again effing off doing who knows what. But today's episode is brought to you by Touching Grass. It's good for your mental health, and it turns out it's also good for the health of your immune system. So we're going to talk about a pretty cool paper about rewilding mice and seeing how that affects their immune system. But first, we've got something a little bit special, continuing our tradition this season of bringing politics into science. We're going to talk with a grad student at Emory University who organized the Atlanta March for Science. So we recorded that previously. I'm going to cut that in here. But it was a cool conversation, and I'm excited to get to it. So before we talk about mice running around in fields, let's go to Jeff Morgan.
Great. So I'm going to just sort of jump right in here. I mentioned that you are a doctoral student here at Emory. And so full disclosure, I am a faculty member within the program that you are a doctoral student in. So we
Nothing you say here will affect your grade, Jeff. Positively or negatively.
We do know each other. But I wanted to start by just having you give us a little bit of background about yourself, maybe who you are, how you found yourself at Emory, and perhaps some of the science that you're involved in here.
Yeah. So I guess I can start back with what made me decide to go immunology. I actually graduated in UC Santa Barbara, so I'm a California boy originally. I actually was in biopsych, which is like some weird combination of cognition and neuroscience. And my focus was primarily on the pharmacokinetics of addiction. That was my favorite thing. But there's not a whole lot of undergrad work, right out of undergrad work in this field. And so I just started looking for lab experience. I wanted some time before jumping into a PhD just to work in a lab and then see what that's like. So this is at the end of 2019. I'm applying, and I managed to get a job down in La Jolla Institute for Immunology with Dr. Sete and Bjorn Peters. And so this is a vaccine discovery lab right at the end of 2019. And we know what happens. Yeah, exactly. We have COVID hit right in 2020. I remember having the whole conversation of, I'm hearing news about this thing. Maybe a couple weeks of this. Maybe a month. Maybe. Yeah, and we know what happened.
It's just in Seattle. That's fine.
Yeah.
It'll stay on the boats.
Gosh, those conversations. It's just funny looking back now, really. And
funny is one word for it.
Yeah, we have to keep light of it, I guess. Being in vaccine discovery I stayed there until 2022. So a couple years of working in immunology, and I was like, you know what? I like this. This has good impact. We're doing things to help people, and I can see myself doing this. Because in the end, studying addiction is also just helping people. Like, that was the idea. So from there, I started applying to a few different universities, and I actually applied to universities and applied to move into the biotech field because I wanted just to move in some sort of direction. And so I just applied to a few of the universities, and my girlfriend at the time, she had family down in Georgia. So I was looking around at universities and saw Emory and went, oh, you know what? Pretty good for immunology. I like these professors. So let's just, send an application to Emory. Sent one to Toronto, sent one to a few UCs. got back some. I got back some acceptances and just kind of compared my experience there as well as what they were offering. And UCSD actually offered less stipend than Emory. And just for some context, I was there for 2 years working as a tech. I got paid less than Chick-fil-A employees. And so
Weird,
I
right?
just, yeah, and I was like, I'm living with 5 other people. We share a fridge. It works, but I'm also 25 and kind of want maybe just a smaller house or something just to share a fridge with 2 other people instead of 5. And so when they offered less than Emory, which is about what I was making then, I went, I know what this means. This will be 6 years. I'll be into my 30s with, the same situation or worse. So you know what? Let's go to Georgia.
I mean, to be fair,
We're glad. Oh, go ahead.
no, I was going to say, places in La Jolla can offer way less because they expect you to be able to, live in a tent on the beach rather than actually pay rent, which is not a bad life. I went to UC San Diego for my undergrad, so I know that area well. It's a beautiful part of the country.
It is, yeah.
Smaller mosquitoes than Georgia, I hear.
Yes.
But yeah, that makes a ton of sense. I think it's sort of funny because I feel like, I don't know if this was true for you, Matt, but I feel like when we were applying to graduate school, there was sort of no expectation of money being, it was like, do it for the purity of the science, which I think is a really toxic attitude to have. And so I really appreciate that you are upfront about making financial choices
That are realistic.
and make sense.
So full disclosure, so Kevin and I know each other well through our own graduate program when we were going through our doctoral program together, and we were in a house of 5 people that right up until about age 30 were sharing one fridge. So
It's
we're very
true. It's
familiar
true.
with that situation.
Absolutely.
Yeah. So, well, we are certainly glad that you found your way here to Emory, and I know that you're working on some cool science now, but what we really wanted to talk a little bit about today was some of the policy that's been influencing the field and science in general over the last few weeks. So some of our listeners know that we've been doing sort of 2 things over the last month and a half, 2 months or so. We've continued to record episodes that are a little more science focused. We did a cool episode on itch the other day. But we also have been starting to record independently some updates on the policy side of science, how it's impacting things at Emory, locally, as well as more globally. Now, Kevin and I obviously are coming at that from a faculty perspective at this point, and you can sort of understand how it's impacting your own program and project how it's affecting others. But I hoped you'd be willing to talk to us just for a couple minutes about what life has been like over the last few months as a graduate student.
Oh, yeah, absolutely. I mean, it's been chaotic, to put it, if I were to just try to say one word on it. We get this news coming at us from everywhere, social media, so much speculation as to what's happening. And I know that this hits, everyone all the way up, professors and deans and everything. And the issue, of course, being that if you guys don't know what's happening, we certainly don't know what's happening. And so I remember having conversations with certain professors who were really on the mindset that nothing is going to impact us until those NIH cuts come out. And then hearing back from that and then going, okay, well, what happens to me as a grad student? Am I moving around? Am I not going to get paid? And so I remember after reading all this, reaching out to the head of IMP, emailing him and saying, look, I'm not the only grad student who's wondering what's happening. And I know you're wondering what's happening. But if you could just give a little bit, a little email to all the grad students and just say, hey, we're working on it, or we don't know either, but your stipends are good, or just something, so that we know you're thinking about it, at least something. And fortunately, we have a great head of IMP who then responded with a email that I'm sure you got as well, Dr. Woodruff. Yeah. So that was just the early times, I think. The other side of things, I mean, early is what, 2 weeks, but
Yeah, right. Yeah, no, it's important to keep in mind that the time frame that we're talking about here is from late January till now, right? Which is an insanely short amount of time.
yeah. And where I was going with that is like the early phase was just a level of panic for me. I remember I had to change some of my habits, like viewing Reddit, for example. That's only in the mornings now. Do not view that at night. You will not be able to sleep. It was my discovery. And so panic and not knowing what I can do, and really thinking about my post-graduation life came up a lot, because I'm not overly interested in pursuing postdoc. There's other, I told you I thought about applying to PhD and biotech. My interest was in industry. And when I came here, it even opened up to CDC. We have a professor here, Bill, who I spoke to. He does a lot of antibiotic resistance. And he got us in touch last year with somebody from the CDC and just had her come in and talk with us about what her job means. And so this was a big thing for me too, where I went, oh, I never even considered government, CDC, NIH. And I asked her a lot of questions, wrote it all down, and was like, okay, you know what, maybe not biotech, maybe not academic, maybe CDC, maybe NIH. And so, and then everything happens, right? And that's when we start seeing this loss of what we hold dear, our institutions that matter so much to us. And for me, it wasn't just that, it's just the loss of, now where do I go? We talking with other grad students too about this, and some graduate students who didn't have anything impact their grants, and they feel like, oh, it's not that big of a deal. And then I tried to explain to them, understand that when you go to graduate, there's less jobs in the job market, but also all those scientists that just got fired from the NIH, that just got fired from the CDC, they're going into the same job market. You're competing now with thousands of other scientists with government experience. You're going to lose.
Yeah, no, Jeff, you bring up a really good point, and it's something that we've discussed a lot. And just as a quick recap for those that have not been following our show or may not be aware, there have been a number of different policies that have come through over the last few months, really sort of all orthogonally directed at really kind of tearing at the sides of academic science, but also, as you mentioned, governmental science, right? So the CDC, the NIH, the NSF have all experienced the kinds of federal layoff conditions that have been seen across U.S. government in general. And as you're saying, I mean, a lot of these folks are really seasoned, and we talked last episode about sort of the push that's going to have to happen into biotech, and whether that space is going to be able to hold those folks and maintain that level of expertise. The answer is almost certainly that it won't. But you're right. It creates a really challenging environment for those in transition periods. And I would say, you mentioned before, it's sort of it hitting all the way up and down the chain, and I think that that's absolutely true. I mean, you're in a stage where everything is in flux, and so the chaos you're feeling is obviously extraordinarily real and impactful. And also, we are in contact with folks that are currently on a faculty job search, for example, when every university simultaneously has decided basically to freeze those searches.
It's not just academia and government science. I mean, the pipeline of research into biotech is huge as well. And due to these policies and a number of others, we're entering an uncertain economy, uncertain regulatory environment. Biotech companies are pulling back too. So I just met yesterday with a friend of Matt and mine from graduate school who was in the virology program. She's on the job market right now and not able to find anything in biotech. She's got 10 years experience since her PhD working in startups, working in really intense research environments in the biotech industry, and she's having trouble finding things because biotech companies are not sure how to invest in their drug development pipelines at the moment as well. So it's not it's we sitting in our perches in academia, we have a set of concerns, but it's broader. It's the entire biomedical research industrial complex in the U.S. Is supported on the back of government funding, and it's
so that paints a really rosy picture, Jeff, and I apologize for the catastrophe we're now raining down.
You're
on
not
everyone's
graduating
heads.
soon, are you?
No. So, but what I would
not.
say is, I think you spoke to this a second ago, but there have been two major responses, I think, to all of this. One has been what I think has been up until this point a pretty reasonable response, which is really keep your head down and try not to make noise and try not to attract attention, and hopefully the funding works itself out, and hopefully the damage isn't too bad, right? I personally am of the belief, which is why we started recording some of these episodes, that that's not going to cut it this time. That it is probably time for folks to stand up and begin taking action where they feel like they can. And so I just wanted to say I really appreciate the work that you did putting together Stand Up for Science, and I wonder if you could talk a little bit about what sort of drove you to start organizing.
100%. I think I'm of the same feelings of you. Out of the lab and into the streets is one of those chants that we had going on, and put down the pipette and raise up a fist. These are the things that we're inside. But this is about 2 weeks ago, where I'm calling senators, I'm calling House members, and I'm just trying to get through to them and say, what are you doing? Trying to push. But I found that it just wasn't effective, especially with some of our senators already kind of support us. And so we're just reaching out to people who are already sort of trying, but they don't necessarily know how impactful this is going to be to all of Georgia and to all of U.S., as we just discussed. We knew.
Yeah, and just as a quick point of context, so our senators are John Ossoff and Raphael Warnock, who, despite Georgia traditionally being a red state, are actually both blue Democratic senators, and in support, I think, of many of the policies we would be supportive of.
Yeah, well, and the other, so we have Governor Kemp, right, who, and our Attorney General, who didn't join the lawsuit to protect the NIH direct cuts, right? These are the two guys that should have had Georgia in on that lawsuit that California and other blue states followed. And so these are the guys that are ignoring their constituents. And so trying to figure out exactly what we can do. I ran across an article. I don't remember exactly what the journal's name was, but it was a grad student discussing this Stand Up for Science 2025 early days, when all they had was a website. And it had no pages, no Eventbrite, it's just a website. And I read the name, and it said Emory University. And I went, oh, I bet I could find this person. So I found this person. I emailed her and said, what can I do? And she said, perfect. I'm going to D.C. Why don't you be, here in Atlanta? And so she had, at one point, she had a post that was just like, we need volunteers to be state lead. We need volunteers to just help organize. I sent her an email with a list of what I've done in the past, which is not protesting. It's more I'm a student representative for IMP. I am an event organizer for IgG. It's just a club for immunology here. So I'm involved in that type of thing, but not activism until this. She reached back out and said, we need to get moving on this. You can be Atlanta State lead. Figure it out. And so I did. It was about... Oh, go ahead.
I was just going to say,
Kevin.
we talk about soft skills you learn in graduate school. It's not only about organizing experiments and writing papers. Turns out you can do lots of stuff with the stuff that makes you successful in graduate school. So that's awesome to put that to use in this context.
And I think you'd be surprised as to how many activism stories start almost exactly the way that you just described. Like, hey, we need this thing done. Can you do all of it? Sure.
Wait,
Yeah.
oh, I guess that is me. Okay.
Oh, yeah. I mean, some background, my lab works with primates, right? We're at the Emory Primate Center, and that's a lot of coordination. And so this is just, like you said, these soft skills that I learned, I put to the test with this. Instead of coordinating with vet staff, I was trying to coordinate with everybody I could get a hold of. And the first part is figuring out how legal can this be. So this is contacting Atlanta Police Department, contacting Georgia Building Authority, contacting Capitol Police. And this was especially important during that email that came out about 3 days before the protest, or 4 days. The tweet from Trump that said illegal protest, which then I had to be the one to email everybody on the Eventbrite and everybody and say, first of all, illegal protest isn't really a thing. But also, I've been going through approvals with Atlanta Police Department, Capitol Police, GBA. We have a reservation. This is, if there's an illegal protest, this is the farthest from that, whatever that might be. So it was a lot of coordinating and a lot of trying to go through these, the hoops, I guess, of just what we need to do, what we need to bring, what's acceptable at this plaza.
Mm-hmm. No, and it's necessary work, right? Because you unfortunately really risk undercutting the message. If you get caught in a situation where you haven't established, right, the licensing to, or the approvals to be where you are, then no one cares, right? You get posted up as another example of some flash protest that shouldn't be there, doing something it shouldn't be. So good for you for going through all of those hoops so that we can sit here talking about the event itself and not the other thing that can come from this kind of effort, to be honest with you. So how did it go?
Yeah, so I think it went really great. When we first, so week one of our big push, which is about a week before the protest, we got the Eventbrite up and started posting. I walked all over Emory and just put up posters, hours and hours, and we got about 200. And so I thought, okay, let's aim for 200. And so I get porta potties set up because we need to have about one porta potty per 100 person. And then the following week comes out, and then we have 300 people, and then 400 people. And so I'm panicking, trying to get more porta potties and trying to get this all set up. And then I get back word, because we're scrambling to get speakers. And so I'm reaching out to everybody I know, and we're trying to spread it and not just have Emory Professor 1, Emory Professor 2, Emory Professor 3. No offense.
No, I get it.
And so
Hear enough of us as it
is. Some of the organizers are from Georgia Tech, UGA, Georgia State University, so I'm having them talk to people they know. And then I reach out to a bunch of professors I know who know people in CDC and say, is there anybody who can talk? I know this is a precarious position. Or maybe instead of have them talk, do they know anybody who got fired who can talk? And so I'm reaching out to these people, and I got in contact with a couple. Most of them were either out of town or not the people working at CDC, the people who got fired, or they're in the job market and they're just unavailable right now. So that part, I couldn't quite get a speaker who was fired from CDC, but the spread starts to move this way to different speakers and everything. And I think we got about 15 people, which range from House representatives, like Dr. Clark, who was there for March for Science in 2017, one of our inspirations, right? We had some early research techs, somebody I know in Mirko's lab who spoke about this impact on her family. We had a lawyer come out who talked about exactly this fight that he's been having with Attorney General, and so as far as speakers went, I was very happy with it. Our chants were sort of on the fly, and I think the hardest part that we had was we needed to keep the sound system to a certain decibel level to be allowed at the Liberty Plaza. So our sound system wasn't loud enough to reach all the 400 to 500 people we had. And I think turnout-wise, I know I got contacted by some CDC people who did say they were going to be present. They just don't want to be known that they're present. Sure. So they didn't sign up on Eventbrite. And so our final Eventbrite numbers are about 400. But we had somebody there counting, and she hit up to 500.
Nice.
And so in comparison to DC, which had like 2,000, I think San Francisco, but we're in Georgia. This is traditionally red, and we've started. And that was what I wanted to reiterate with this whole protest that we had, was this is actually just a start. This isn't like now everything's fixed. Ta-da. This is more so get like-minded people here and start building connections.
Absolutely.
And I think it's happening. We had people from 5051. Mm-hmm. They came and talked to me afterwards and said that they want to collaborate. We had somebody who has a dancing goats right up in Buckhead who came up and he said, I'm an owner of it and I want you guys to come here and we'll support you and give you space to do whatever you need to do. So it's the start, which, it's exciting to see. And I'm still within the Nation organization. We have a Discord that we use. And we're throwing out ideas left and right of what we can do to continue to advance this, and we're capitalizing on that. I think something that was quite effective that we did in Atlanta that not all the states did was we had a follow-up QR code that was just posted around the protest that you can scan and give us feedback. And we had about 100. So maybe 1/5 of the people scan this and give us the feedback, which we were looking for. Yeah, and a lot of them said, I think 80 people said that they wanted to continue to help and wanted to know ways they could.
Great.
And they were really interested on legislation. And I actually have it pulled up right here so I can just take a look at my responses too. The top ones, 92% said, I'm worried about how funding cuts will negatively affect all of us in the future. Mm-hmm. So, this is why they came out. This is exactly what we were looking for. And the top hit was contact legislators, selected officials to have public science funding and advocate for science is what they want to continue to do. So we did what we wanted, which is start.
Yeah, that's
Good.
great. And I got to say
That's excellent.
a couple things. One, so I was at the Boston protest, Stand Up for Science, and you are not the only one that had issues with sound system. It was very hard to hear anything on the Boston Common, so don't feel bad about that. I think the follow-up thing is great. I got handed a flyer there. That's the only sort of outreach that I got, and I think after reading it, I was like, this organization seems sketchy, so I'm not super into it. Anyway, I think so. I think the follow up is great, and I think you're absolutely right. Like building organization, building groups of people that can fight. Like things are moving so fast. I think
They are.
having the organization and starting to build up. The resistance is a sort of damaged term now, but I think building up organizations and groups of people to advocate.
Right. Solid advocacy.
Yeah.
Yep.
Yeah.
So, great. Well, you've told us a lot, I think, about the Stand Up for Science, and obviously things are continuing to move forward. I wonder if there's anything else that you wanted to share with us.
Yeah, absolutely. I want to give a shout out to the Stand Up for Science Georgia group, like the group of organizers that helped with all of this. Without their contribution and help, there'd be no way that we would have had that protest last week. They did a lot of reservation, talking with media, and ensuring that we would have basically a protest together. So I definitely want to give a shout out and props to all of them that helped me out with that. Yeah.
Great. Yeah, as you mentioned before, a lot of logistical planning that goes into this kind of thing. So it's wonderful that you have the infrastructure for that.
Oh yeah. They're great. I mean, they've come from Georgia Tech and GSU and all so it was just very exciting to be able to connect with other universities on this organization level, and just see what they're able to. It's kind of like being in a group project where everybody does their part or more so. Everyone's contributing 110% in this group that I'm a part of. So it's been really smooth to be able to just have somebody who I know. I say, like, this is the thing. You want to do music. You're bringing on musicians. Can you handle that? And then all done. Don't have to think about it. Fully taken care of.
It's an amazing feeling, isn't it?
It is. Yeah.
Excellent. Well, hopefully you can keep that momentum going. It sounds like you're generating some pretty good practice.
Yeah. I'm sure. I hope we can too. Yeah.
Excellent. If folks are looking to get involved, how would they go about doing that? How would they get in touch with you?
Cool. Yeah.
How would they get in touch with the organization?
So I think one of the most direct ways will be to email standupforsciencegeorgia@gmail.com. So this is our current inbox that we're using, and this will go straight to me. And so this is like the most direct way to get involved, because now I can start plugging you into things or contacting you. And one of the things that we're currently working on is to combine some level of outreach with privacy, which is very difficult. But we're ironing that out right now. We have some ideas on moving to Proton email and moving to what is the name of that?
Matrix
starts
chat?
with the S.
Or Signal?
I can't think of it.
Signal.
Signal. And so these communications, especially for our Fed family, right? They don't want to get too involved with Google. But besides that, you email us and we'll do our best to try to coordinate. And that's the most direct route.
Cool.
The other means, of course, is we've got another one coming up here, a protest at CDC literally tomorrow. So I don't know when this will be posted. Maybe it'll be
Probably
too
not by
late for
then,
that one.
but
yeah,
we can
yeah, fair enough.
send us the info, send us links. We'll stick it in the show description. Share it on social media, and yeah.
Yeah, I'll start sending things right over to both of you guys. I sent out a follow-up email to all the Eventbrite signups, and the emails that responded to the QR code and said they wanted to help. How?
Awesome.
And so I can send it to both of you as well, and just see where I can't say that this is nationwide exactly what Stand Up for Science is doing, because they're coordinating right now. The head. Organizers. But I know within Georgia, we're kind of quite active And so we're trying to quickly capitalize on this and get movements going and get people to continue to stay. Just the momentum, keeping the momentum
Absolutely.
going.
And I think doing this in places like Georgia that are not deep blue, like Massachusetts is, California. I feel like is among the more important things. So yeah. So amazing work, and really great to have you talk about it.
Thank you once again. Thank you. I really appreciate it. And I got to say, what you guys are doing are great. This is exactly something that science can sometimes have a difficult time with, which is getting what we are doing in our university out to people to see what's going on.
Absolutely.
And so this is great. Thank you.
Yeah, cool.
You're very welcome. Thank you again for joining us, and hopefully we can get some of that done. See what we can do.
All right. Thanks, Jeff.
Excellent. Thank you so much.
All right, we're back. So that was great. Jeff seems like an amazing student to have. I don't know how much lab work he's getting done, but seems like a good thing to have people who are passionate about.
I imagine more than you'd expect.
That's true. No, I mean, regardless, I think it's great to have somebody that's so dedicated to making science work. Because, like, if everybody keeps their heads down in labs, like, we're not going to have a science community to do anything. So I think it's really valuable work he's doing.
It'll make this podcast real boring, real fast.
Yeah. Well, there's a lot of history we could get into.
It's possible that our production value would go up because it'd be literally the only thing we'd be doing.
That's true.
So we just
So if you want to make sure that Matt and I can continue to do this when we're not getting paid, you can go to support us on Patreon, patreon.com/audioimmunity. We're now pulling in, just from our last episode, we're pulling in a sweet $3 per month.
Woo!
We're rocking. If you care to support us, you can go to Patreon. You can, kick us $1 a month, $2 a month, whatever you think is appropriate to the work we're doing here. And you can also leave feedback on Patreon. Tell us what we're doing right, what we're doing wrong. If you like all of this, policy content, or if you would like us to shut the hell up and just talk about science. Although I shouldn't say it that way, because this is science in 2025. Yeah. This is, we sort of can't get away from it, unfortunately.
Yep. So we're going to keep doing that. But we'll warn you maybe before we start doing it. Although I would say that our recurring What's Good in Academic 2025 segment is a good way to know that we're about to jump down that rabbit hole. So
Yeah, I think there is a way to do chapters on podcasts, and I've been thinking I should figure that out so that I can add chapter markers to make sure people can skip ahead if they don't want to hear about us griping. Although this conversation with Jeff I thought was really inspiring. It's kind of amazing to have people doing that work. So
yeah, it's cool to see people just jump into advocacy because there's an obvious need to be filled, and even if they don't know how they themselves are going to get it done, they just start doing things. And I think you brought up during the interview, and I think it's a good point that folks that are successful in science and academic science actually bring a lot of skills to the table. It's not just sort of science at the bench or your hands or any of those things. There's a lot of critical thinking. There are a lot of organizing skills, as Jeff mentioned, that really lend itself to a lot of different potential avenues to get involved. So I guess I would encourage folks that are out there listening to this, maybe you're in science, maybe you're not, but you almost certainly have a tool set that if you find the right organization will allow you to get involved and have a meaningful impact. So really it's often just about the activation energy of trying to get over that hump of what am I doing? Do I belong here? The answer is yes, you do. Just start doing it, and people find ways to help.
Yeah, I have to say, it would have never occurred to me to order porta potties. If I was organizing a rally, that would have just never occurred to me. Good on him for figuring that stuff out. But yeah, so today we're going to talk about a paper that's called Rewilding Catalyzes Maturation of the Humoral Immune System. And the first author, I think there's two first authors here, Yihan Chen and Kimberly Zeldana, and looks like two last authors as well, Sergey Korolov and Ken Cadwell. So I saw this pop up on Blue Sky, and it sort of reminded me of some of the conversations you and I have had about genetically outbred mice and how using inbred strains can cause all kinds of weird stuff to happen in the immune system. And this paper is about inbred strains that are just not held in the facilities that we tend to imagine.
Yeah, it's really a cool paper, actually. We'll get into some of the difficulties in doing what they're trying to do in a little bit, but you're right. We've had a lot of these conversations, I think some on this podcast, that like genetic inbreeding generally frowned upon in humans, right? The reason for that is that you develop these recessive traits through families that when you're nicely mixed in with a genetically diverse population tends not to rear their heads. But when people or animals or anything really starts to breed into a smaller and smaller genetic pool, those recessive traits have a tendency to show up. And often they're not great. We've talked about the susceptibility, I believe, of the Cavendish banana in podcasts long ago, where essentially every single banana tree is susceptible to exactly the same disease because there is no diversity in the ability to fight off incoming pathogens. Obviously, it gets more severe than that. You just have to look at the royal lineages across Europe in the Middle Ages to identify some of the interesting traits that came out of people routinely marrying their brothers and sisters. So not something that we advocate.
First cousins.
And first cousins.
Not ideal.
Yeah, not
And
great,
the reason,
but
just to put a finer point on it, the reason for that is because we all carry around, I forget what the exact number is, but it's something like a half dozen. Everybody has like a half dozen recessive alleles that if you were homozygous, in other words, if you had two copies of that allele, you would have problems of various sorts. And, under most circumstances, in an even reasonably diverse population, the mix of those recessive alleles that are deleterious is wide enough that you're only going to inherit like 3 from your mom and 3 from your dad on average, if it's 6. Again, don't quote me on the numbers here, but they're not going to be matched up. But if you have what we call consanguineous populations, in other words, sharing blood, lots of interrelations, you're increasing the probability that you're going to get 2 copies of the same deleterious recessive allele. This happens in some communities that are that tend to be more insular. So there's one example is the Ashkenazi Jews, partly as a result of bottleneck because of the Holocaust, but also because it's sort of an insular community that more typically marries within the same religion. You get that. There's a number of examples in modern times. Like I think the Saudi royal family, there's a lot of sort of first cousin marriages there, and actually in Boston, Boston Children's Hospital is one of the best places to get care for a bunch of these sort of uncommon genetic disorders that are the result of this interbreeding, and of course the European royal family in the Middle Ages, as Matt was mentioning. But that's sort of just wanted to sort of clarify the reason for that is really just like random mutation causes deleterious mutations. If you are marrying, if you're having offspring with people that are closely related, more likely to get those overlaps.
Right. And so that's, of course, when we decided that we were going to start studying immunology. That is exactly what we did. What we took was we took a bunch of wild mice and started breeding them, and then we bred them again to each other, and then we bred those again to each other. And we kept doing that over and over and over again. And the result of that were the strains that most immunologists now know and love. So there are a whole bunch of them. And the interesting piece about them is that they all, or at least the ones that we talk about routinely, BALB-Cs, Black 6s, J1s, they don't behave immunologically the same. Now, it's not always clear why they don't behave immunologically the same, but they frequently don't, which is why we've discussed in the past, I believe on this show, about the potential benefit of using genetically outbred mice. That's something that my lab does increasingly, and Kevin, you and I are working on a little bit of that together, and there's some really interesting stuff that happens to an immune system when you actually start adding in at least a taste of the genetic diversity that might be out in the wild.
Smidge.
A mild amount. And also, despite all of that, despite the differences that we see in those genetic backgrounds, there is still this huge factor, which is environment. And we have talked a lot about this over the course of producing this podcast over the last 11 years or whatever it is. Your immune system is a pretty good fighting force, but that's not what it's for, really, right? This is something that we go back to all the time when talking with students that are sort of approaching the immune system for the first time. It's not wrong to think of the immune system as a defense force in certain circumstances. But in reality, a significant majority of what your immune system is doing at any given moment is really maintaining balance between a highly, highly diverse and changing world on the outside of your body. And you have to match that with a similarly diverse and flexible system on the inside. Right? So we often talk about your intestinal tract, right? So you've got feet and meters and feet of bacteria. And
I thought you were going to talk about the microbiome on the soles of your feet for a second there. I was confused.
No. But that is an interesting thing, too. It turns out that probably in each one of your fingernails, for example, you have a different microbiome. And the interesting piece about how flexible and how diverse these systems are is that your immune system is not ignorant of any of it, right? We have this idea of immune-privileged spaces that sometimes comes up in immunology, which is almost certainly not true in the way that we traditionally think about it. But these aren't even immune-privileged spaces, right? Your fingernail beds, your teeth, your intestinal tract, the difference between your small intestine and your large intestine and your colon, right? All of those things are highly diverse.
I mean, even distal colon and proximal colon are vastly different, yeah.
So in all of those spaces, your immune system is present and paying attention to what's going on. It's not ignorant to the bacterial colonies that are growing and feeding off each other and fighting each other for control and getting parasitized and being, in these complex predator-prey relationships. It sees all of that. And its job is not to stop it. Its job is to monitor and sort of garden that normal flora so that you can have a productive interaction with the outside world. We've talked before about how bacteria are a critical component of our own human metabolism, and that doesn't work without bacterial colonies that are healthy and not killing each other all the time, right? And your immune system...
Matt, are you saying that we need to change the tagline of our podcast to a podcast about our body's never-ending attempt to reason with the outside world?
I believe peacekeeping is perhaps we could consider it. But yeah, I think that this interaction between our immune system and the world around us is really what defines its function on a day-to-day basis, right? And we've seen this pop up in a lot of different ways, right? If you change the microbiome of an animal, it changes the way things like autoimmune diseases play out. So substantial changes in gut bacterial composition can have impacts on the way your brain is attacked by your immune system, right? These things are all connected, they're all dynamic. and highly complex. And so one of the things that we often overlook when we're doing these immune studies is that all of our mice, regardless of the genetic background, the strain, all of them are in the same, generally, specific pathogen-free facility, right? So what does this actually look like? And I'm sure I've described this before on the podcast, but basically, if you go downstairs from where I'm sitting, 3 floors...
It's always in the basement.
And that's important, right? Because you actually have to control the light. You have to make sure that circadian rhythms are carefully controlled so that, animals don't get out of whack. Out of whack is a technical term for some reason your vaccination didn't work because you woke up a mouse in the middle of its sleep cycle, and that actually turns out to be important. So when you go downstairs, like I said, about 3 floors from where I'm sitting, what you'll find are rooms that have card swipe access and big heavy doors. And those big heavy doors are so that we contain, right, everything that is going on inside that room into that room, right? You can imagine that airflow is tightly controlled. Interestingly, going back, all of this is dependent on those indirect funds that we talked about before, maintaining this kind of infrastructure. But essentially, you tap into these rooms, you open the door, there's a whoosh of air that goes into the room because they're all positive pressure, and you walk in, and what you're confronted with is a number of racks of cages. And so you can
you missed the fact that you've gowned up, you've put on little booties over your feet, you generally put a hairnet on.
Yeah, no, that is true for many facilities. Not all, as it turns out, but many facilities. You go in and you have these large, I think what could be best described as sort of pet store looking racks plugged into central water lines, right? And so you can monitor the animals for health. You can obviously take individual cages in and out. You can do your experiments. But the point of the room being set up the way that it is because there are a lot of pathogens, as you know, that can infect mice as well as humans. And some of the mouse pathogens are particularly infectious within these genetic strains that we traditionally use. I studied way back, God, I was like 8 or something. No, I think that I was on a summer internship at the New York State Department of Health. This probably would have been my sophomore year of undergraduate, right? So we were studying Ectromelia virus. And Ectromelia virus is one of the specific pathogens that a specific pathogen-free facility is trying to control. And the reason for that is that if you accidentally release Ectromelia virus into a mouse colony, all of them will die, right? Like it'll basically rage through like smallpox. It's actually a pox virus, which is,
Or like
bad.
measles, maybe?
It's bad.
Another
for
topical.
Mice. Yeah, it's bad for mice. So I was not allowed to go anywhere near that facility. I was not allowed access to it. I had to stay 50 feet from the door or something like that. Like they were really cautious when I was going that I would not be interacting with that colony because there is really no resistance once some of those mice get infected for it to spread rapidly through the entire colony. So we are on the lookout for those bacteria, viruses, pathogens generally because we don't want to kill all the mice in our colony. We don't want to make them sick. It interferes with the immune system. But when we do that, we are also restricting the amount of diversity that these animals can possibly be exposed to, right? So if your immune system is highly dependent on the outside world, right, your microbiome, in order to develop properly, as we were just talking about, in order to do, 95% of its function, and you bottleneck the diversity of the normal flora by routinely going back to these specific pathogen-free facilities, it stands to reason that you might be mucking around with the ability of an immune system develop, quote-unquote, normally even in mice where you've introduced lots of genetic diversity, you are still not recovering sort of a normal natural phenotype because you haven't actually allowed them to go out into the world and experience the diversity of, pathogens, fungi, all of those things that mice get exposed to. And that gets to our paper, which you mentioned is called Rewilding Catalyzes Maturation of the Humoral Immune System. And so I want to describe the methods right up front on this, because I think it's fun. Basically, what this team did was they developed an outside cage, which I love, right? I imagine me owning a hamster at 6 years old, like putting a cage around a part of my yard and letting the hamster run free, which I definitely did. That's basically what they've done here. So what they describe is essentially releasing these 6 to 8 week old mice into the yard, wherever that is, and letting them hang out. So they interact with bugs, they interact with the grass, as Kevin mentioned, they interact with dirt, presumably. They just kind of live their mouse lives for a little while.
I am presuming they don't describe this. It might be in their previous paper because this is not the first paper where they use this system. But I was wondering, like, it's got to be covered, right? Because they don't want birds of prey coming in, I would guess. They do specifically mention that there are no other mice. There are no wild mice in this habitat.
Yeah. And they say it'd be interesting to see what the actual setup is, because they say that, that's to prevent pathogens from getting in and out. It strikes me that perhaps that's not sufficient to allow pathogens not to get in and out, but
but you're not going to have like nose-to-nose mice,
right?
With wild mice, right? So you're not going to have like direct contact, but there's going to be lots of soil bugs. They mention a couple of like more like opportunistic pathogens like Staph aureus that are kind of like pathobiont is a term that's sometimes used where it's like could be a commensal, but sometimes it's not. Sometimes it gets real bad. So those things are around. But yeah, I just kind of like. I'm wondering, like, do they have like a courtyard? Like, if you imagine like where the Modell Center used to be in at Harvard Medical School, like they just have like a courtyard inside of a building. Do they just like let the mice go run on the grass there? Or
it feels right to me. I would mention that in the methods they do talk about pathogen screen to make sure that that at least select pathogens have not found their way back into the colony. I imagine if I'm thinking about IACUC protocols and divisions of animal resources, thinking about that experiment, I imagine this is a nightmare to get approval for. Right?
Yeah, IACUC, the International Association for the Care and Use of Animals or something like that. So it's like international body that sort of governs ethical treatment of animals. I mean, to be fair, like I think this is more ethical than keeping mice in cages their whole lives. Like even if they are going to get picked off by a hawk, I feel like it's much nicer for them to be able to run around.
No, I think you're right. I think the problem they would have is all the other animals in the colony, right? Like, what are you bringing back into that colony, and how are those? How is that going to be handled and cordoned off from everybody else? So, anyway, that is the premise of this paper. They're going to take mice. They're going to look at them in a lab setting. They're going to let them go out into the world. Different cohorts, not the same animals. Let them go out into the world, recapture them from the world that they've been released into, and then take a look to see if the immune system has changed.
Can you just imagine the conversation between those mice? Like, oh, we had it so good, and we fell for the cheese again. Damn it.
Yeah, I mean, I don't know what the recapture looked like. There's always that.
I feel sad for the mice that they get back to their cage in the animal facility, and they're like, "Damn it, Joe! I had it so good. I had it so good out there in the world."
I do want to emphasize in the middle of this that we do take very good care of these animals in a lab setting. So we're making light a little bit here, but I do want to assure everybody that like that the reason that IACUC committee exists is because there's extraordinary scrutiny over the housing of these animals, how they're treated, every single procedure that they go through, all of those things. But also, it would be sad to have a taste of the outside and then sort of be drawn back into the lab life environment.
Another thing that just occurred to me, actually, I don't think I read in the methods what the sex of the animals they're releasing. Because, these are mature mice. Mm-hmm. Are they making sure that the facility only has one sex so that you don't get breeding? Because mice breed pretty fast, and they're pretty eager to do it. So
they do. And if you fail to recognize that a mouse is pregnant, your assessment of their immune system is going to throw you off. In the previous study, both male and female mice were analyzed, and the impact of sex on immune variables was shown to be minimal and not significant for lymphocyte numbers. To prevent breeding and aggressive behavior in the outdoor enclosure, only female mice were used for rewilding in the subsequent experiment, while both sexes were included as laboratory controls. So their laboratory animals were both male and female. All of the mice that they released out into the wild were female, which is an interesting component, although they assure us that has nothing to do with anything that we're about to see. So here's an interesting thing about B cells. Couple interesting things about B cells. We know B cells are antibody producers, right? So let's just start off there. B cells grow up in the bone marrow, right? When they are first minted, a B cell has on its surface a B cell receptor. That B cell receptor looks a whole lot like an antibody attached. I'm doing it for YouTube. Attached to its surface like this. So it has one grabby on either side, right? It looks like an antibody because it is an antibody. It's basically an antibody that is membrane bound into the outer membrane, and it actually doesn't signal directly, right? What it does is it associates with some other signaling molecules. So if something binds to that B cell receptor, then that B cell sort of senses that the receptor has been bound. Now, importantly, every B cell has exactly, at least under canonical understanding, every B cell has exactly one kind of antibody out on its surface, right? One kind of B cell receptor. So each individual B cell has its own sort of identity as to the range of molecules, proteins, whatever, that it can bind to. Right? And there is incredible diversity across B cells, right? So we've got, tons and tons and tons of different individual B cells, all with slightly different affinities to different kinds of proteins, and they all just float around, right? And they wait for their B cell receptor to see something they might be interested in. And then you go through this downstream differentiation process where you go from what they call a naive B cell that hasn't yet seen anything it recognizes to a fully functioning antibody producing, what we call antibody secreting cell, right? So a cell that is specialized in the production of antibody for protection of the host. Now you can also become a memory B cell, which maybe secretes a little bit less antibody than an antibody secreting cell, but nonetheless still probably contributes to the humoral immune response. So humoral immunity is what we refer to when we're talking about antibody-mediated immunity.
Because it's from the humors, obviously.
Right, of course.
Vitreous humors.
Yes. And so those memory B cells are responsible for maintaining the memory of the humoral compartment, right? So imagine you get the flu, for example, and you have a bunch of B cells that respond and they say, oh, look, my B cell receptor binds to flu. So you take a couple weeks, you generate a bunch of antibody secreting cells, you generate a bunch of memory B cells, and hopefully you fight it off. Hopefully you're not dead. Hopefully it's not avian flu and now we have another pandemic. So you survive that fight, right? The flu goes away, you clear it, no more flu, right? Couple things happen. One, is that most of the B cells that form the underpinnings of that humoral response, most of them are going to die. That is just normal contraction. We talked about that before. Every immune system that expands eventually has to contract, or else you become a big ball of immunity and then you die. So most everything around that humoral response goes away, except for a couple populations. One are long-lived antibody-secreting cells, and those have a tendency, at least canonically, to be understood as going to the bone marrow. They set up residence there, and they can live a very, very long time. So when you think about people that were vaccinated against smallpox, right? So vaccinia virus vaccine recipients. Those folks, 60 years later, 70 years later, continue to have antibody-secreting cells from that vaccine that are hanging out in their bone marrow, presumably secreting antibodies and preventing them from getting reinfected.
Well, and
Which
folks
is cool.
that were alive during the 1918 H1N1 pandemic, in 2009 when we had the swine flu H1N1, turned out that those people, 90 years previous, they still had memory responses to H1N1 flu.
Yeah, so we have two kinds of responses, right? The first is any existing antibody that continues to circulate because those antibody-secreting cells are sitting in the bone marrow and spitting out antibody. And also you have a circulating memory B cell population that if it sees a new pathogen that it might recognize, even if it's not exactly the same, but it can recognize enough of some protein, right? So in flu, let's say hemagglutinin has changed significantly since the strain you saw, but it's close enough where you can still sort of get a handle on it. Those memory B cells can start ramping up their antibody production a lot faster than a new naive B cell response, right? So this is the way that we think about B cell immunology. We think about naive B cells. Prior to that, really transitional B cells coming out of the bone marrow, turning into like fully formed naive B cells that are circulating but haven't yet seen their antigen. At some point, they hit an outside antigen, and they generally will develop into this response that will eventually become either a short-term effector that will die, a memory B cell that will continue circulating, or a long-lived antibody-secreting cell in the bone marrow, right? So that is our B cell paradigm. Now, there's some other pieces of that developmental process that we'll get into in a bit. But here's the problem in studying this kind of thing in mice. The problem is CD27. You might not know what CD27 does. So CD27, it's not clear to me, actually, what it does. And the reason it's not clear to me is it's one of those immune markers that I never cared enough about to actually figure out the function, because what we use it for is a differentiation factor, right? CD27 is extraordinarily important in B cell biology in general, because in human beings, CD27 is a responsible, repeatable, confident marker of a memory B cell population, right? So if you're looking in human blood, you can gauge on some basic things that tell you, yeah, this is a B cell. We tend to use things like B220 and CD19 and, some other things that doesn't actually matter what they're doing, but they're telling you it's a B cell. If you see CD27 on its surface, it is a pretty good indication that that cell is a memory B cell. And that includes antibody-secreting cells. Those are also CD27 positive. So we're actually leaving behind a number of cells that are a relatively large population of cells that my work during COVID was really focused on. But by and large, the way that we understand B cell immunology, particularly in humans, is through this paradigm where we take B cells, we separate them by the expression of IgD, which is a flavor of B cell receptor that is relatively immature-ish, and then by CD27. And that tells us whether or not the B cell has matured through affinity or through class switching by removing IgD from its surface, turning its B cell receptor into more functional downstream antibodies, and also whether it has gained sort of that memory marker. This is why that's a problem. Mice don't have CD27 in any meaningful way, it seems. CD27 does not mark memory B cells in the blood in mice in the way that it marks it in humans. A further problem is, I don't know if you know this, but mice have less blood than you do, right? So humans have what, 4 to 5 liters of blood? Human adult, I think it's somewhere in there. Mice have 4 to 5 milliliters of full-grown mouse, right? So we're talking 1,000 times less blood that you could potentially draw out of a mouse, which means one of three
To be
things.
clear, you're never drawing 4 to 5 liters of blood out of a human.
No, nor
just to
would
be
you ever
clear.
draw 4 to 5 milliliters of blood out of a mouse. No,
No,
yeah, we're
no.
talking about total volume. So you have 3 options, really. If you are studying blood B cell populations in mice. The first is you can take really small blood samples and figure out how to do cool biology using really small blood samples. And Kevin, you know this, but this is something that as soon as I got a lab, we started figuring out, because one of the goals of the lab is to sort of study mouse blood in the way that we study human blood, which is longitudinally and individually based, right? So we wanted that sort of kinetic assessment.
Matt did
The
an
second-
internship at Theranos, so he decided
Right.
that. No, I'm just kidding.
That's exactly. No, exactly. And if you have venture capital...
That's not
funding-
true. I made that up, just to be clear.
Yeah. So you can do that. You can take really tiny blood samples and see what immunology you can do. Or you can do what most folks did. You can do what these folks did, which is do terminal bleeds, which means that you sack the animal at whatever time point you're looking for, and you get as much blood out as you can. Usually it's a mil if you have a good operator, maybe a little bit more than that.
And to be
probably
clear,
up to
for
2, but
for people that aren't into the jargon, we're not putting mice in sacks. Sack is short for sacrifice.
sacrificing.
So we're
correct.
killing the mice. Yes.
Correct, yes. The mouse is sacrificed and we draw as much blood as possible. That is how most of these experiments are done. The third way to go about this is don't study the blood, you idiot. Why would you make it hard on yourself? Nothing cool is happening there anyway, right? B cells aren't developing in the blood. The immune response isn't developing in the blood. We've talked about how all of the adaptive immune response is functionally happening in lymph nodes and spleen and sometimes in peripheral tissue. So what do you even care about blood for? You fool? And to that I say perhaps. But also,
Could
the
be.
problem with that is that we have built up an extraordinary amount of information about how B cells operate in human disease conditions. Now, you can't get lymph nodes and spleen routinely from people. There are some new techniques that allow you to actually take some of those biopsies, which is kind of cool. But still, you're not going to routinely be studying the developmental sites, right, in a person, and that is highly related to disease state, right? Like you want to study B cells in the context of COVID-19 and sepsis and HIV and all of these things. And so what you're going to routinely have access to in humans is the blood. So those things have to marry, but without CD27, it is extraordinarily hard to line those two things up. And all that is to say that we're going to dive into this paper and we're going to run into that problem again and again and again. What do we mean when we say an immature versus a mature versus a naive B cell? What do we mean when we say a memory B cell, right? Actually, the thing that is most consistent between mice and humans are the antibody secreting cells. They pretty routinely express at least some of the same markers that allow us to fairly rigorously identify them and track them. But everything up until that phase is actually much more nebulous than you'd expect because we just don't routinely do blood studies in mice of B cells. Right?
I just want to pause here to say I am so extraordinarily glad I asked you to do this paper because I started looking at these figures and I was like, markers. I vaguely remember these from graduate school. Ah, crap. So I'm very glad that I don't have to go through and explain what all of the CD44 and I mean, I remember what MHC2 is, of course, but most of these markers are just like 2 or 3 neurons firing somewhere in the cortex being like, that sounds familiar, but I have no idea what they are.
Yeah, and I mean, I'm going to leave most of that alone because, to be honest with you, I think that, we could really get into the weeds. But here's what there is to know, right? What there is to know is that different people, good immunologists, have fundamentally disagreed as to what these markers mean, right? There's a lot of literature in a lot of different directions that tries to explain B cell biology based on sets of markers that are presented to the community. And probably all of them have some level of validity, right? Probably all of them have shortcomings, right? It is simply that because nothing as clean as CD27 has emerged, that there is just sort of a back and forth on, what all of this really means. But we'll get into that a little bit perhaps as we go, but we should start by saying basic method here. Take a mouse, put it in your backyard, wait a while, grab the mouse back, sacrifice the mouse, take the blood compartment, run it by flow cytometry, so a really dense set of potential markers that you're looking for, so you can break it down into B cells and T cells and all of these things. And then compare that to the mice that you did not release into your backyard. And so one of the first things that they saw was that a marker that is often used for T cell activation, CD44, goes up, right? So in the blood, we tend to use CD44 as a way to differentiate memory T cells. It's way cleaner than what you're showing here. Right? Like when you do this in T cells, you can actually take naive T cells and reasonably well separate them from effector T cells, for example. Here, what we're looking at is a bump, right? We've got an increased bump of CD44 positive B cells, looks to be about twofold. Twofold is a lot when you're talking about blood composition, especially of live lymphocytes, although that's within the B cell population. So, you're getting an expansion, I guess, of CD19 positive B cells in general. And then also within that B cell compartment, you seem to have this CD44 expression. So that is essentially where the whole paper starts. We let these mice out into the world, we brought them back, and when we brought them back, there are more B cells. And there are more B cells that seem to be expressing this memory marker CD44 or activation marker CD44. But there's an important component to that. Remember that we're in the blood, right? Everything that we're talking about here is in the blood, I think.
For those of you not on YouTube, Matt just leaned over to his computer to double check that he's not full of crap.
Right.
I think in this figure it's all blood.
Yeah, we don't get to lymph node until E. So we are still in the peripheral blood. There are no B cell T cell interactions that we know of going on in the peripheral blood. Right? So this represents less of a B cell that is actively talking to T cells in the environment and more of a B cell that you'd expect would potentially be capable of talking with a T cell, right? And
so are these B cells generally like the CD44 positive? Are they just like moving between lymph nodes in the spleen looking for T cell help, or are these like on their way to some tissue? As effectors, or
so I don't know the answer to that, and I think that this is why a lot of this is up to interpretation, right? Like, what does it mean to become a mature B cell in their way of sort of phrasing it, right? What
you've
is
had
the
your bar mitzvah and are ready to be
Yeah, well,
Just
but
kidding.
I mean, but it's an important point, right? Because what are we talking about here? Are we talking about B cells that have seen something specific to their B cell receptor and are now upregulating CD40 and MHC class 2? Probably not, right? Or else the percentage of CD19 that are MHC class 2 positive wouldn't be near 100%, right? Not all of these mouse B cells have experienced antigen, right?
Why do you think that's the case? That these wouldn't mostly have seen something specific? Because a lot of B cell receptors are for random microbial sugars and are, there's going to be a lot of. We've already talked about how these rewilded mice are going to have a lot more exposure to a lot more microbes. Not that these are all pathogens necessarily, but it doesn't strike me as impossible that there's a lot more B cells circulating in the blood because they are constantly encountering the whatever external signal that they're specific for.
Yeah.
Is that crazy?
It's not entirely crazy, right? So what you're talking about is tickling, right? So you're not getting an actual full-on binding event, because what we know is if you get a full-on binding event, that cell takes action, right? It's not going to stay in the peripheral blood just hanging out wondering what it's supposed to do, right? It's very likely going to try to find its way into a secondary lymphoid organ. It's going to try to find a T cell that can provide help, all of those things. So what we could be looking at here is sort of a tickling event, certainly. So some sort of breakdown of the gut barrier that allows more microbial components to find its way into the blood. Maybe there's more sort of quick on-off binding and low affinity interactions. That's possible. Another possibility is that your inflammatory systemic environment has an impact on the way these B cells operate at a fundamental level. And I would argue, based on what we know in humans, which is that if you take a foundationally autoimmune inflammatory person, you can see changes in the transcriptome all the way back into the developmental phases of those B cells, right? So it seems like the microenvironment and the inflammatory environment has a pretty profound effect on the biology of these cells. So best interpretation is that what we're looking at here is the difference between a B cell population that is really quiescent in a really sort of low inflammatory state and B cells that have sort of been poised to become more active players in a new onset immune response, right? So we can test some of that a little more directly by looking at the developmental environments, right? And so this is where we start to get into the question of what is an immature versus a mature B cell. And here we're looking at a couple different options. One is the presence or absence of CD93. They make the argument, as others have, that if you're expressing a lot of CD93, you might be more immature. If you're expressing less CD93, you might be more mature. The balance there, again, is sort of tricky. We're not yet talking about B cell receptor mediated activation, right? We're just talking about final maturation into what would be considered a resting naive B cell. Same is true of CD23, right? You're trying to sort of use CD23 as a proxy for maturity versus immaturity in these populations. In both cases, what they show is that these markers that have been published and validated in other people's labs, looking at sort of the development of a mature naive B cell, those markers seem to show you that if you rewild these mice, they transition from what is a relatively immature phenotype into a relatively mature phenotype. Okay, obviously characterized by slightly increased expression of CD44, CD40, and MHC2. Now there is an interesting piece. So we go through We talk about immaturity based on those two markers. And then we look a little bit at IgM and B220. And this is the first spot where we have to be a little bit careful when we think again about mice versus humans. An interesting component of mice, and Kevin, you know this because I showed you some primary data, but there are actually two fundamental populations of B cells in mice. They both circulate, although in black 6 animals, one circulates at a much higher frequency than the other. B2 B cells, right? What we call sort of follicular B cells. These are the routinely circulating B cells that we think about when we think about canonical immunology. Those form sort of the backbone of the circulating B cells that exist in a mouse. But there is a second population of B cells, B1 B cells. Those B cells are largely spleen resident, but we've seen in certain strains can actually circulate at quite high frequencies, right? So these are cells that are a little more, you can think of them as innate-like. Their receptors are a little more genetically constrained. They have a tendency to be express higher reactivity to normal flora, right? So like these natural antibody responses, they tend to be more poised towards that. They tend to be a little more autoreactive. They tend to develop fewer mutations, for example, over their lifespan. So these are cells that are not frequently seen in the blood, but we know from other genetic outbred studies that it is possible for these cells to circulate at much higher frequencies than we normally see on a black 6 background. The reason that that's relevant here is because when we're looking at this recirculating versus immature population that they've gated here, the things that change between a B1 and a B2 B cell are the levels of CD19 expression, B220 expression, and IgM expression, right? You can actually pull those populations out based on those 3 markers in a reasonable way. And so we have to be really careful here when we're talking about recirculating versus immature that we're not accidentally gating on a changing B1 versus B2 B cell ratio. That said, let's take it at face value, right? I think that the majority of the evidence in this figure tells us that we are probably getting a switch in phenotype from sort of a more or a less inflammatory to a more inflammatory milieu, right?
And that's
All
figure
right.
1.
Cool, that's figure 1. So then the question is, are we actually generating new responses? Are we causing these B cells to get activated? We've talked about the developmental environments of these cells previously. Generally speaking, when we think about B cells turning into these long-term antibody secreting cells or memory cells, they have to go through this training environment, a germinal center environment. And we've talked a lot about this before, but in that environment, B cells do a couple things. They change their receptors to hopefully develop higher and higher affinity to the antibody they recognize, or the antigen they recognize. And also, they make sure that the antibodies they're producing are not self-reactive, right? So there's a series of checks and balances that goes into the production of these things. And so they go into
Checks
the
and balances.
lymph nodes.
That seems like a good thing. Maybe we should have that in our government.
Yeah, we'll see. So what we can see is...
Sorry, I didn't mean to bring it back, but it just...
No, it's okay.
You use that phrase a lot, and it's been on my mind a lot recently, so just had to call it out.
Yep. No, I get it. I was trying to gloss over it without commenting myself, so it's fine. So what they're looking at here now is we're looking at lymph nodes and spleen, and we're really asking the question, what kinds of activity do we see in these secondary lymphoid organs? Do we see evidence of increased germinal center formation? Do we see evidence of increased antibody secreting cell formation? Right? What are we seeing? So in panel C, we're taking a look at the spleen and we're asking the question, do we have more germinal center B cells, more cells going through this selection process? And the answer is, yeah, sure. Like, I would say that if you were looking at a raging infection, or if you were looking at a vaccine delivered into the blood, or, really any strong stimulation, it would look much more resolved than this. This is a pretty moderate phenotype, but yeah, I think we see some increased germinal center activity.
But I mean, that's
in this
we
spleen.
wouldn't expect to see evidence of infection because they're sort of explicitly trying to keep these mice away from active like pathogens that have evolved to infect mice, right? So this is still, I think, it makes sense. It's more evidence that there's just like a generally more mature and active immune system without it being like we are responding to something acute, right?
Yes, potentially. But I want to draw your attention to G, right? Which is the number of plasma cells in the spleen, which is way higher, right? We've got a pretty good statistical outcome there. And then look at the bone marrow next to it, right? So we said that as you're developing, you have a couple different things that can be going on, right? You can develop into a short-term effector and produce a bunch of antibodies. You can become a memory B cell, or you can become this sort of long-lived antibody-secreting cell, long-lived plasma cell, right? So what they're showing you between G and H is that the number of antibody-secreting cells in the spleen is going up, almost double. Pretty good result. Not so much in the bone marrow. Can you say that there's something going on there? Probably, right? Much more sort of muted phenotype. And interestingly, if you go into the mesenteric lymph node, right, in J, you've got a pretty strong result there too. There's a lot of class switching to IgA, which are B cells that are producing antibodies specifically that can cross the gut mucosal barrier, right? So they can affect
and mesenteric lymph node
and
just
control.
Right. For the non-aficionados, mesenteric lymph node is the lymph node that drains the gut and the
Right,
gut lamina
exactly.
propria.
Exactly. So these would be responses hopefully aimed at controlling whatever's going on in the normal flora in the gut, right? So what we're seeing here, I think, altogether is, are we seeing some germinal center response? Maybe a little bit, right? But we're actually seeing a pretty robust effector response without an enormous amount of long-lived antibody secreting potential. And this is the discussion that I said we would come back to about how you become a fully functional B cell. So I mentioned that I had done some work in COVID-19, and a lot of that work was in evaluating non-canonical pathways or less canonical pathways of B cell activation that do not go through this germinal center response. And it turns out you can actually get the production of antibody secreting cells and effector B cells without going through a germinal center, right? And those cells tend not to turn into memory. They tend not to turn into antibody-secreting cells that are long-lived, that engraft into the bone marrow, but they do exist. And so looking at this data, you have to sort of ask the question, is there a possibility that you are getting the effector development of these cells without necessarily establishing a full-on antibody-secreting cell response? I think the answer to that is probably yes, but it is admittedly hard to get into those pathways in the blood, particularly if it's not your very specific field. So what's going on with these B cells? So now we're going to do what we always do, which is turn to single cell analysis. We have chosen 4 animals.
It's so hot right now.
I feel like hopefully it's dying back a little bit, but I know that that's not true. Like I want it so badly to just pull back a little bit, but we're not there. So what they did was they took the peripheral blood of 4 mice, 4 lab mice, 4 rewilded mice, right? And basically did a PBMC analysis by single cell transcriptome. And what you see in this figure is a flow plot, I think. Mostly. So what we've done here is we've taken our transcriptomes, and in A, we have used all of those transcriptomes to create a UMAP projection Presumably there was a PCA step in there somewhere, and now you're doing UMAP projection of all those cells that you captured by single cell. And then you're using a library of transcriptomes that allow you to take those cells and predict what kinds of cells they were based on their full transcriptomes. They show you and see some of the genes that make them confident that they are in fact those cells that they called. And then, honestly, in D&E, it's kind of like a reworked flow plot. So if you had a dense enough panel, and to be fair, you can't always put together a 40 color panel to get every single subset of cells. So like innate lymphocyte type 3s might be hard to get into your flow panel if you're trying to capture everything. But basically what you're seeing in D is that by single cell, they see an expansion of B cells in the peripheral blood of rewilded animals at the expense of, it seems to me, CD4 helper T cells, right? So CD4 helper T cells contract. Plasma cells, interestingly, contract from the blood, but they showed that that's not the case in either the spleen or the mesenteric lymph node. And really, we have an expansion of B cells. Now, we shouldn't really see germinal center B cells. This is a thing that infuriates me. Germinal center B cells should not be in the blood. Unless, if you're listening to this and you disagree with me, and you believe that germinal center B cells are in the blood, I would like you to email me and please explain to me why a germinal center B cell would be in the blood. Because I have not figured that out. It finds its way into the literature now and again. I don't understand. But that's basically.
Are there not germinal centers in the blood, Matt?
As far as I can tell, no. So, yeah.
Are there not these structures that are really dependent on the stroma of a specific secondary lymphoid tissue? Is that not happening in the blood? Do we not have little clusters of B and T cells?
As far as I can tell,
little
what you're
follicles
describing,
swimming through the blood.
I believe what you're describing is an embolism. And those generally are not conducive to animal health. So, to be honest, and this is a thing that plagues immunology in general, is we assume that markers that we use in lymphoid structures are in the blood because they're left over from when those cells were in the secondary lymphoid structure, right? So if you use CD95 and GL7, for example, to gate on germinal center B cells in the lymph node, which we do routinely, and then you see those same markers in the blood, you say to yourself, oh, that's a germinal center B cell in the blood, assuming that the B cell has ejected itself from the germinal center and has accidentally found its way into the blood without downregulating those markers. And I think that is probably the wrong way to think about this most of the time. We do this all the time with chemokine receptors. We assume that the production of or expression of a chemokine receptor is reflective of past biology rather than current biology. And it drives me up the wall, to be honest with you.
Well, I mean, this is part of a larger problem. I'm sure that I have ranted about this before, but of the fact that we are using, traditionally cell surface markers, but in this case, like specific gene expression patterns, to give names to things.
Buckets.
The names of which refer to some aspect of biology that can be very misleading. So the favorite example of this is the plasmacytoid dendritic cell, which is so dendrite is like a long branching extension. Plasmacytoid means spherical, and so these are spherical dendritic cells, and it's all based on this marker, CD11C. Yep. We don't have to relitigate that, but I think this sounds like a very similar thing.
Just
like
did.
We're not litigating. We're not litigating.
Okay. Just stating the truth of the thing.
Just stating that this is a thing that is dumb. And yes.
Fair enough. All right,
and immunologists are particularly prone to it. It happens in other parts of biology as well. But I think the immunologist reliance for so long on flow cytometry makes it a particularly acute problem in our
Yeah.
particular scientific lineage.
And often for good reason, because you're trying to separate out very complex things, and so you have to be able to name things, and you run out of ways to name things. So you start to refer to markers, and then you become dependent on the markers, and then the marker changes, and it's a mess. It's just hard. It's complex stuff.
Yeah.
So, speaking of complexity, why do a UMAP on all of your cells when flow cytometry could have done? And the answer is because you are looking for heterogeneity within specific subsets that you may not be aware of, right? So you do this single cell experiment not to separate T cells from B cells, but to understand the differences between B cells in rewilded versus lab mice. And so that is what we are doing in figure 4. We're taking that same data set.
Well, before we get there,
Yeah.
just real quick, I found this very confusing. Figure 3, the title of figure 3 is Rewilding Leads to an Expansion in Intestinal B Cells. Most of figure 3 is about this single cell experiment, not intestinal B cells, right? Oh no, these are cells isolated from small intestine.
Yeah.
Oh, I missed that.
Yep.
Okay, excuse
Yep.
me. So this single cell experiment was specifically from intestinally isolated B cells.
It occurs to me that everything that I just bitched about germinal center B cells not being a thing, is not correct.
Because this is not in the blood.
Because this is not in the blood. No, you're right. No, the cells here were isolated from the intestine. And germinal centers can form in tertiary structures in the intestine. So that would be less problematic.
Are Peyer's patches
Nonetheless,
considered tertiary or secondary?
Peyer's patches.
I thought Peyer's
believe,
patches
are
were considered secondary lymphoid structures.
Yeah.
Yeah.
But you can also get tertiary lymphoid structures within
Right.
the gut mucosa, which would have, I think, a GC-ish phenotype. Okay, so erase the germinal center conversation. Still, what we have done here is we've isolated cells. We've shown that there are more B cells in the intestine, fewer CD4 helper T cells. Yeah? I apologize to the authors for mischaracterizing that through most of that last conversation. So, nonetheless, we still could have arrived to that result with flow cytometry, but where single cell gets interesting is you can then dig into the differences between cells, finer heterogeneity. And so what they're taking a look at is they're looking at really what they're classifying as 3 different kinds of B cells. They're looking at plasma cells, so antibody secreting cells. They're looking at general B cells, and they're looking at germinal center B cells, even though germinal center B cells were a pretty small fraction of their overall B cell composition actually in both the rewilded and the lab animals from the previous figure. And so we're looking at some changes in proportion. Largely in the lab animals, we're seeing a decrease in antibody secreting cells, plasma cells, from the lab to the rewilded. We're seeing an increase in mature B cells into that environment. And a slight, we're talking 1.6 to 2.3% increase from the lab to the rewilded group. Not clear if that is statistically significant. My guess is not, or else they would have shown that it was, and these were 4 mice per group. So I think what we're seeing.
Yeah, you
is a
can't
trend
really
across
do
those 4 animals.
Yeah, you can't really do that kind of statistic on this. Do they even are they even
You
separating
can
out the 4 different?
If it's real tight. I think
Yeah.
that they're not specifically
I think they're
because
just-
the N on those cells is going to be too low.
Yeah,
Yeah.
right. They're pooling all of the samples, so you don't even have error bars here. You just have the number pooled across the 4 animals.
Yeah, so we, in some of the COVID work, we would do work on like 4 or 5 people, and you can do it, but you've got to have consistent biology, and you've got to have a pretty good number of cells coming out of this. Which leads to, unfortunately, a really unclear assessment of the diversity of the antibodies and B cell receptors in F So I'm going to skip over E for a second. But another thing that you can do when you're assessing B cells by single cells, you can actually look at the receptors themselves and ask the question, are you getting more diversity in the B cell receptors of these rewilded animals versus lab animals, right? You might expect that as you increase the diversity of the normal flora, that you'd expect an increase in the diversity of the B cell receptors. And the answer to that is yes. Not super clear, to be honest with you, what I'm looking at there, other than yes, it does appear that the alpha diversity within that B cell receptor is slightly increased.
So to spell this out, so this is because we're not just doing like PCR to measure transcripts, we're actually sequencing transcripts. We then use that sequence to assign it to a particular gene. But for B cells, depending on your sequencing depth, you might have reads that are crossing those variable portions of the antibody gene. So from the transcript, you can sort of say what the sequence is. I didn't dig in to see like how deep the sequencing is. So how many reads would I expect to land on these variable regions of the B cell receptor? I was sort of shocked that this was like even technically possible. So yeah.
Yeah.
I don't know.
I mean, it's not so hard if you're getting good coverage. I think if I remember, they had a couple thousand receptor sequences per group, which is not bad. What you find in a situation like this, and I don't know if they trying to find here. I imagine this was all sequenced in bulk. Yeah, so we successfully captured 1,381 and 1,178 productive BCRs in laboratory and rewilded mice. What you'll find.
Okay, so you're talking about across all of your B cells.
Yeah.
Irrespective of what stage of maturity.
Yes.
We're talking about like in the neighborhood of 1,200 individual BCRs.
Yeah, which is really problematic, actually, for reasons that... so if you pool B cells and you say, I'm going to do B cell repertoire sequencing, what you get is you get a representation of the antibody secreting cells.
Right.
The reason for that is antibody secreting cells are secreting, or they're producing like 10
Yeah, they've got way more transcripts, right?
10 to 100 fold transcripts, right? So what you get is a huge dropout problem where you're thinking that you're getting an unbiased, right, representation of the entire B cell population. Really what you've done is you've sequenced the antibody secreting cells, and I'm sure they know that here. They're not using any of the designations for B cell, germinal center versus plasma cell here, I imagine.
Well, I feel like you couldn't, again, because the numbers are going to be so small that you're going to get like 10 transcripts from a germinal center B cell or whatever. So it doesn't make any sense. Anyway, that's...
Yeah, and
one of the many reasons that doing relative abundance kinds of things from sequencing, especially if your sequencing depth is necessarily low, because these experiments are expensive, it's just, I feel like it's hard to say anything about it.
What they do say, and I guess, gives you some measure, so a little more than half of the total B cells in the lab mice would have had to have come from plasma cells based on their numbers here. Actually, plasma cell would have been a lower contribution than rewilded, but basically what they show is that if you actually do look at antibody-secreting cells, the mutation frequency is not so different, although they're a little less mutated maybe in the lab strains, but not clearly different, I would argue, between the two. They make an argument that there's some change in the IgD versus IgA usage frequencies. Again, I mean, maybe, it looks like it, but without animal replicates here, you don't know if you've got an animal, especially in the rewilded group, that has significantly different biology that you've compiled here. It's very hard to know what's going on. Nonetheless, moving on, they do something actually that's pretty interesting in 5. And that's where we'll finish this paper up, which is to ask the question, are you getting changes in the targeting of microbial communities by these different populations? Now, for reasons, well, basically what you're doing is you're extracting bacteria from fecal samples, and you are taking the IgM or the IgG component from the serum of those animals, and or plasma, I guess, and you're asking the question, does the IgM and the IgG enrich for specific bacterial populations now, right? And how does that look in a rewilded setting versus how does it look in a lab setting? You can't do this with IgA because there's tons of IgA in the fecal sample directly, right?
Yeah.
So
though I have to say, when I was first looking at this, I was like, that's weird. They're doing IgG and not IgA, because there's a more and more common technique now called IG-seq, where you just, from a stool sample, you just pull down all of the antibody-bound bugs from the IgA that had been secreted into the gut. So you could absolutely do that. It's not this mixing of serum and stool the way they're doing here, but that's kind of weird anyway. Why wouldn't you take the antibody that's already in the stool that has been secreted specifically to target the microbiota and then look at that? But that's, that's fine.
Yep. Yeah. So we're looking at IgM and IgG. IgM being sort of the more immature version, usually lower affinity, usually higher avidity, because IgMs have this sort of like concatemerized structure that they form. And so they ask the question, first of all, are there differences in the microbiota composition between rewilded and lab animals? And I think you'll agree, based on C and the PCA, the answer is yeah. Yep, you do get changes in colonization. So then they took their IgG-bound bacteria and took a look at those in rewilded and lab, and it does appear that the rewilded animals, interestingly, seem to get less diverse. Is that fair? So the IgG seems to almost concentrate the signal a little bit, or there's more diversity in what the IgG is targeting in the lab animals, I guess would be a better way to put that.
Yeah, I mean, the problem that I find with these plots, and even with D to some extent where they're trying to quantify it using this scoring methodology, is that the input microbiota is so different.
Yeah.
That it's hard to say. It's hard to say how much of the difference that you're seeing in the antibody-bound fractions is a result of different immune responses versus a difference in just the input composition, right? It could have been kind of cool to say, we're going to take serum from lab animals and try to bind the stool microbes from the rewilded or something like that. I still don't know exactly what that would tell you, but this whole figure, to me, I mean, it's interesting as a descriptive thing, but it struck me as a reviewer asked for kind of figure. I mean, even though it's the last figure of the paper, it's compared to the detail of the other stuff, this feels a little bit half-assed to me. And I mean, maybe that's just because I'm, my bias is a microbiome person now, but I don't know what to make of this. It's just kind of
Kevin.
They have different microbiomes. That is not shocking. They're in wildly different environments. They have different microbiomes.
Yeah.
What does that mean? I have no idea.
Yeah, and I would, sort of moving in, basically what they're showing is that, there are seeming differences between, for whatever reason, the rewilded versus lab animals that are bound, both by the IgG and IgM. You can separate them out by PCA and C. And then if you ask the question, are there specific bugs that IgM and IgG are binding more, for example, in the rewilded mice, The answer is yes, especially on the IgM side. There seems to be a fair amount of new binding to new kinds of flora that are captured in the IgM. Interestingly, you don't see that in the IgG. There doesn't seem to be a lot of sort of new emerging IgG targeting of specific bacteria, at least within the class switched compartment, which is maybe not entirely surprising, because what we're talking about here is, so IgG responses, right, are sort of dedicated to systemic, more
Right.
Th1 driven, non-mucosal kinds of infections. I'm not sure that you would really expect a lot of the difference here to be in the IgG, and in fact,
No, that's exactly right.
the
You'd expect it to be in the IgA compartment. Exactly. And so
Right.
I was sort of interpreting this as being like the IgMs are like those more naive B cells, right? IgM is the baseline isotype prior to class switching. And so I was sort of thinking like these are the ones that are being expanded, like getting ready to class switch to IgA, where they're going to secrete it into the gut. So I would expect if they had done like more traditional IG-seq, where they pull down the IgA from the stool samples and sequence that, like I would have expected to see something more like what we're seeing on the IgM side, maybe even to a greater extent.
Yeah.
But yeah, it strikes me as weird that like the fact that you are mixing serum, like why wouldn't you do the cross experiment where you take lab housed animals, and you use their serum to bind the microbiomes of the rewilded mice and vice versa?
Yeah, I think
I don't know.
that would help in interpretation. I agree with you.
You couldn't do that part with IgA-seq when you're pulling down from the stool sample, because you can't isolate the IgA
Right,
effectively
and then
from the bugs.
pull it out and then cross it. Yeah.
So,
Yeah.
I don't know.
Yeah.
I was very put off by this figure, to be honest.
you seem put off. You seem mildly irritated. So I
True.
mean, I think that's really what we got. So here's my general view of the paper. I think it is interesting to document changes in B cell phenotype. I think that we've known that there is a potential for environments to play a large role in the potential downstream immunology that we see in vaccination, infection, and all of those things. I think that it's very clear that that keeping mice in a very contained environment, and certainly in a genetically inbred situation, constrains the amount of biology that we're able to see, because the farther you get away from a human setting, the harder it is to interpret. I think that they have made a good effort here to try to incorporate a fairly confusing literature base around the maturation of B cells to come to the idea that that it does feel like these cells are hitting a more mature sort of posture out as they're circulating around. I think that they're seeing probably expectedly an increase in antibody secreting cells throughout the system, specifically in the intestine, which is good, makes some sense. It's not clear to me that what they're seeing is largely germinal center derived. I think it could be. It could be that what we're seeing is germinal center derived purely. My better guess is that there is a sort of extrafollicular component here that is probably playing a role and creating these sort of short-term antibody secreting cell effectors, which is why they're not seeing a substantial increase in the bone marrow. But it could also be that they're just not waiting long enough. And, if you came back, a year from now or something like that, you would see that. And beyond that, I mean, the transcriptome analysis that I didn't hit very hard, honestly, didn't reveal all that much. Beyond the surface receptor changes, I think that, they've got some changes in the metabolism, which are always good to sort of document in an immune context. It's not always clear what it means when a metabolic state shifts. Do they have access to different metabolites in that environment because the microbiome is different, because they're in a different bacterial setting, or is there something changing about the activity of these cells that switches the way that they rely on glycolysis versus other pathways, those kinds of things? It's sort of hard to say. Nonetheless, I think the overall broad strokes take home is that indeed, if you take mice and you put them out into your yard and you bring them back later, maybe a little sadder than they were before. They probably,
or happy because they had the opportunity. You know, we don't know.
Maybe, yeah,
we don't know.
We can't measure the gratefulness of mice.
It's true.
It's challenging.
Not yet.
Not yet. But
so
we do see a change in B cell posture, orientation, baseline resting state.
So I have two questions for you.
Yes.
The first question is, do you think that there's a T-cell paper forthcoming, or do you think that the T-cells just weren't that varied and so they focused on the B-cell? Because to me, if I were doing this, I would think that this is like a pretty descriptive study, which makes sense. This is like a quite a novel method, and so like descriptive paper makes a ton of sense here.
Yep.
I feel like if it were me, you've got the serum. Like, why wouldn't you do all of the immune compartments that you could plausibly look at in the blood? Mm-hmm. Why would you focus so much on the B cells?
Yeah, I mean, I think you would focus on them because they said that was the largest, that CD44 shift was the largest signal they saw. Now, I would say that you've got single cell data, which looks like B cells are expanding at the expense of CD4 helper T cells. So in my mind, there's probably a data set out there somewhere looking at the potential changes in the CD4 helper T cell populations. Yeah, I mean, they threw a lot of mice at this study. Like, if you look at, I mean, one of the nice things about working in mice is that usually in a genetically inbred mouse, you can get away with like 4 mice per group, right?
Right.
You've got experiments here where it looks like we're talking about probably 50 animals rewilded, and that's not insignificant, right?
Yeah, no, I mean, it makes sense too, because the error bars, one of the other benefits of being genetically inbred and all, co-housed in SPF is the error bars tend to be a lot tighter.
Yes.
Than for humans, certainly, but also for genetically outbred or apparently for rewilded mice. So, like, you kind of need those numbers to be confident in what you're seeing.
So I guess that would be my answer. My guess is that they've looked at it, whether those error bars are tight enough to actually say anything about the T cell population based on what you're seeing with the B cell signal. Maybe. I would want to get as much out of this data as possible. So
yeah, exactly.
Yep.
All right, so then my other question is, what do you think are the prospects of using rewilded mice to look at things like autoimmunity? Because like one of the major hypotheses around autoimmune disorders, particularly the increasing prevalence of them in the Western world, is the hygiene hypothesis. We've talked about this before. The exposure to more diverse pathogens, particularly bacteria, as you are growing up, matures the immune system in a way that is protective for things like autoimmunity. And so I'm wondering, taking some of our autoimmune models, whether that's EAE or lupus models, different genetic backgrounds. I mean, I don't know how valuable that would be given the expense of this kind of thing. But yeah, what do you think about the prospects for this as a method?
I think that it would take a lot of really dedicated infrastructure to pull this kind of thing off. So I think that if you're thinking about that, the way to do it would be to, I would believe, contact Ken Cadwell or Sergei Korolev about potentially making use of their facilities. But if I was to plan that, what I would do is I would probably be looking for a sort of later onset, slower onset autoimmune disorder So NOD mice, for example, non-obese diabetic mice, develop spontaneous autoimmunity over time. That's just a known feature of the strain. So I think what I would probably do is use an animal like that, just house them outside, protect them from birds, and see if the incidence of autoimmune onset decreases, changes in some way, and I would maybe pick a couple different strains like that and just have a colony ongoing, because it feels to me like most of the acute models in autoimmunity are hammers. So we do something called pristane injection. So think about taking a bunch of insoluble petroleum byproducts, and I don't use this model because I don't understand what's going on with it, but imagine if you just sort of like inject that into the peritoneal cavity of a mouse and then just wait for a bit. What happens is its immune system just freaks out and eventually will start breaking down, right, and start attacking your own tissue. And I would say most of the EAE model, right, where you're sensitizing,
you're injecting myelin with adjuvant.
Exactly. You're intentionally causing that mouse to break tolerance. I think those kinds of things, you'd have a very hard time seeing a difference just because the normal flora change, because these are genetically targeted, often inducers of autoimmunity. Right. That said, these sort of more casual, your immune system is kind of predisposed towards autoimmune breakdown. I think that would be where the money is there. It would take a while. So I guess it depends on how much it costs them to keep mice outside and healthy and away from birds and not dead from other infections. Because then you're talking about
hopefully they didn't use the word diversity in any of their grants. But they're paying for this.
That's right. There's no diversity in B cells. There is only heterogeneity.
Hetero, that should be okay.
What's that?
Is it hetero? That should be okay.
Oh, that's a good point. Yeah. We need to find, so long as they're not transdiverse. Yeah.
Or transgenic.
Yeah. And I just got depressed again. Thanks, Kevin.
That's what I was going for. This has been Audiomunity. I'm Kevin Bonham with Matt Woodruff, and thank you for listening. We are on the web at audiomunity.org. That's AUDIOMMUNITY.org. There you can find links to our podcast RSS feed, links to our Patreon, patreon.com/audiomunity. If you want to kick us $1 or $2 per month, help support what we're doing here. Right now we're doing this as a labor of love. There are a few costs associated with it. It would be great to defray those, but also just, to let us know that you appreciate what we're doing. You can also leave us a comment on Patreon. We don't have a contact email or a form set up, but hopefully we will in the future. The music at the beginning and the end was composed by Rachel Reinick. And I think that's it. We'll see you next time.
Sounds good to me.
Sometimes you get lucky.